To the untrained eye, fragmented skeletal remains appear as silent artifacts, but at a microscopic and chemical level, they are highly structured data recorders. The professional mandate of an ABFA-certified forensic anthropologist is to discard investigative guesswork. They approach the skeleton as a ledger, utilizing rigorous empirical methodology to read information recorded inside. When analyzing a subject, investigators extract 3 distinct measurable data layers from the human bone matrix. This extraction is governed by the strict ethical standards of the Scientific Working Group for Forensic Anthropology, or SWAG-ANTH, which require practitioners to prioritize verifiable scientific accuracy over speculative narratives. The core challenge of any investigation is transforming a decomposing unknown variable into a mathematically and biomechanically verified identity. To do that, the anthropologist isolates the 3 variables of the matrix. The first layer is the chemical signature. This allows investigators to mathematically determine the geographic provenance of the individual. The second data layer is environmental taphonomy, which measures how external forces break down the body to calculate the post-mortem interval. The medico-legal system requires the synthesis of these 3 variables to isolate the forensic signal from the surrounding environmental noise. We begin with the chemical layer. Human bone tissue, specifically the collagen fibers and the mineral bioapatite, acts as an internal isotopic ledger, detailing where a person lived and what they ate. The environment dictates this signature through a process called isotopic fractionation. During photosynthesis, stable carbon-13 and carbon-12 isotopes react at varying rates. Different plant types process these heavy and light isotopes differently based on the local climate. Temperate C3 plants, like wheat and rice, discriminate more against the heavier carbon-13 isotope, creating a distinctly lower delta-13C value. C4 plants, like corn and sugarcane, found in hotter, arid climates, absorb more carbon-13, resulting in elevated delta-13C values. When humans consume these plants, or the animals that eat them, the body routes these molecules with precision. Dietary protein embeds directly in the bone's collagen matrix. Meanwhile, blood bicarbonate routes to the bioapatite mineral structure, capturing the total diet. To test if these culturally specific dietary signatures could identify missing personnel, the Joint POW-MIA Accounting Command Central Identification Laboratory, or JPAC-CIL, analyzed bone samples from U.S. and Asian populations recovered from historical conflict zones. This histogram displays the collagen delta-13C values from 30 of those test samples. The data falls into a strict bimodal distribution. The left cluster isolates the Asian origin group. Their C3 heavy diet, largely dependent on rice, yields a mean delta-13C value of -19.7 per mil. The right peak isolates the US origin group. A high consumption of C4 resources, specifically corn and sugar products, pushes their mean delta-13C value up to -17.4 per mil. This second chart plots the bioapatite data. As you can see, the exact same twin peak separation mirrors the collagen findings independently. Because bioapatite records the entire diet rather than just protein, there is a broader spread across the x-axis, but the separation between the 2 demographic groups remains distinct. To utilize this data in the field, forensic anthropologists apply linear discriminant function, or LDF, analysis to these exact histograms to draw a sharp mathematical boundary between the populations. The algorithm successfully classified the origin of the samples with a 96.7% cross-validated accuracy. These algorithmic predictions were subsequently tested against mitochondrial DNA haplogroup extractions from the same bones, which independently verified the isotopic results. By isolating microscopic chemical ratios, stable isotope forensics enables investigators to definitively separate local indigenous remains from U.S. service members decades after the individuals died. The second analytical layer moves from the internal chemistry of the bone to the external environmental forces acting upon it over time. This is known as taphonomy. Decomposition is not a random process of decay. It is a sequence driven by specific ecological variables that can be empirically measured and calculated. To gather this data, universities operate outdoor research facilities, commonly called body farms. These secure environments provide controlled, longitudinal taphonomic data on human remains. Through this research, forensic scientists identified the three primary variables that dictate the rate of decay: ambient temperature, soil moisture, and aerobic or anaerobic states. These variables are synthesized into the vast postmortem interval formula. This calculation provides law enforcement with a timeline based entirely on environmental inputs. Temperature is the primary catalyst. The formula utilizes a critical threshold set at 12 degrees Celsius. Crossing this line into a warm environment shifts the rate of biological breakdown, increasing the speed of tissue loss. In an aerobic or oxygen-rich environment, the formula maps the total percentage of soft tissue decomposition to the corresponding stage of decay, mapping out the progression from early bloat to active decay. Conversely, flipping the formula to an anaerobic state, such as a burial or tight wrapping, recalculates the timeline, as oxygen deprivation limits the bacteria driving tissue breakdown. The formula operates within strict constraints. The calculated value represents a maximum time if pliable soft tissue remains, and a minimum time if the subject is fully skeletonized or mummified. By relying strictly on observed, quantifiable temperature and moisture data, the formula removes subjective guessing from the establishment of a death timeline. Deriving a mathematical postmortem interval acts as a chronological gatekeeper. It allows investigators to instantly rule out historical artifacts when searching for forensically relevant modern cases. The third layer transitions from the time frame of decay directly to the terminal event itself, focusing purely on the skeleton's physical morphology. The primary investigative challenge here is distinguishing the trauma that caused the death from the postmortem taphonomic damage that occurred decades later. The SWGANT standard dictates that perimortem trauma, injury occurring at or around the time of death, is defined strictly by the biomechanical fracture characteristics of fresh bone. This relies on a fundamental shift in material properties. Living bone exists in a viscoelastic state, allowing it to bend before breaking. Dry, aged bone is highly brittle and stiff. The definitive marker of perimortem trauma is the presence of plastic deformation without any osteogenic reaction or healing. Because fresh bone is viscoelastic, it absorbs kinetic energy. The resulting fractures propagate smoothly along the pathways of the bone cylinder. Compare this directly with postmortem damage. Dry bone lacks a plastic component, resulting in angular, delaminated breaks that snap rigidly under pressure. Environmental indicators confirm this taphonomic breakage. Differential staining, where the newly exposed fracture margins are distinctly lighter than the aged exterior of the bone, proves the break occurred long after death. Only after the timing is scientifically established as perimortem can the anthropologist classify the mechanism of trauma using pattern recognition. High-velocity projectile mechanisms, like ballistic impacts, leave distinct signatures, such as concentric beveling on the cranial vault that points to the exact direction of the internal or external force. Blunt force trauma leaves a different pattern. Rapid deceleration or direct strikes cause contiguous serial fractures and localized crushed bone. Identifying perimortem plastic deformation isolates the kinetic force of the lethal event from the subsequent damage of the environment. Synthesizing these three data streams provides the resolution necessary to resolve the specific identities within commingled mass fatality incidents. In the JPAC-CIL PACE study, investigators faced highly fragmented remains recovered from an aircraft that crashed into a mountain in Vietnam in the mid-1960s. First, PMI calculations established that the environmental exposure of the remains matched the historical timeframe of the specific crash incident. Next, biomechanical morphology revealed plastic deformation and high-velocity blunt impact fractures. This confirmed the trauma was consistent with a crash event rather than later ground taphonomy. Finally, collagen delta-13C analysis isolated the isotopic signature of a single U.S. service member from the commingled local ARVN personnel. Throughout this process, the anthropologist is bound by the SWAG-ANTH Code of Ethics. They must never overstate certainty, and they do not determine the ultimate cause of death. Their role is strictly to read the data, providing the rigorous empirical framework upon which the medical legal system relies to close a case. Human bone functions as a precise mathematical ledger, yielding its record to the correct scientific algorithm.